All right, folks, so our restriction digest is incubating, but we're going to use this time to start getting prepared for the electrophoresis portion of the experiment. One thing that happens pretty often when you're working in a molecular biology lab is you run into situations where you have an incubation or a reaction that takes an hour. It's not really a good use of your time to just wait around while that happens. So while our DNA is incubating with the restriction enzyme, we'll get the gel set up. I have the electrophoresis equipment set up in front of me. We have our power supply that will pass current through the gel once it is put together. We have our gel tank, which is where the electrodes from the power supply will clip to, and it will be full of running buffer. And we have a plastic gel support where we will cast our gel, and a comb that we will use to make the wells in the gel. So what we'll be pouring into this is hot melted agarose in TBE buffer. And you can see that this gel support is open on either end, so the first thing we have to do before we can cast the gel is to tape up the ends so the gel doesn't run out. So I'm going to get a piece of masking tape. That is about a little bit longer than the end of the gel support. I'm going to put that masking tape along the bottom of the gel support so that it overlaps around the edges and underneath a little bit. This will keep melted agarose from leaking out. And I'm going to make sure I seal it really nice and tight. And just to be careful, I'm going to put a second piece of tape across the top. I'm going to do the exact same thing on the other side of the gel support. Going to press that tape down nice and tight, and I'm going to again wrap it around the edges and underneath so that we get a nice tight seal that prevents any melted agarose from leaking out. Get a second piece of tape across the top. So now we have our sealed up gel support, and we can pour our melted agarose into it. I'd say this is probably cool enough to handle, but I'm going to use the green hot glove just to make sure I don't burn myself. At some point, someone told me the dad joke version of this, which is about the hipster molecular biologist who burned their hands because they tried to pick up their gel before it was cool. So I'm going to carefully pour this melted agarose into the support. I'm going to not go super fast here because I don't want to introduce any bubbles. And I'm making sure there's nothing under the support because I don't want anything to make it uneven. So I've poured all of that melted agarose in. And quickly, before it starts to polymerize, I'm going to put in our comb. And our comb is what's going to create the wells that our DNA gets loaded into. So each of these teeth of the comb is going to create an impression in the gel that we can load DNA into. There are two sides of the comb. We're going to use this top side of the comb where the teeth are a little bit thicker, so they create a deeper well. So we're going to put this in just by plunking it down into these slots at the top of the gel support. And I am going to look closely just to make sure that there's no bubbles in the gel. It looks pretty good to me, so I think we're ready to go. This will probably polymerize in about 10 to 15 minutes, and as soon as our DNA is out of the incubation, we'll be ready to load it. Okay, folks, so our DNA has finished digesting and our gel has finished polymerizing right here. So now I'm going to show you how to load your DNA into the gel. So we're going to start by taking our digested DNA sample that we have right here and adding some loading dye to it. So this loading dye accomplishes a few things. It contains some glycerol, which will make the sample, when it's mixed with it, a little bit denser than water, so it will tend to fall down into the well and not immediately diffuse everywhere. It also contains two different colored dyes that migrate with a specific molecular weight. So that will allow us to track the progress of the gel as it is running. I should be clear though that this dye is not the ethidium bromide stain that actually stains the DNA. So we're not staining the DNA here, we're just adding some color to the sample that lets us track the gel as it's running. So I've set my pipette to 2 microliters. I'm going to get a pipette tip. I am going to take up 2 microliters of my loading dye. You can see it is very darkly pigmented, so it will stain my clothing if I'm not careful. I'm going to add that loading dye to my sample. And I am then going to give my sample a quick spin in the microcentrifuge just to make sure that the dye and the sample are completely loaded and spun down to the bottom of the tube. Okay, so that's my sample, it's ready to go. Now it's time to prepare my gel. So I'm going to take up my gel that is polymerized, it's cooled down to room temperature, and the first thing I'm going to do is carefully peel off this masking tape on the ends. If the masking tape were to stay on, we wouldn't be able to efficiently pass an electrical current through the gel and the whole thing wouldn't work. I am going to be careful while I'm doing this to not tilt the gel too much because sometimes the gel can have a tendency to slide out of the support. Okay, so our gel's ready to go, and I'm going to place it down into the tank. So I'm placing it down with the tank electrodes on the right and the ladder, or the comb up at the top. And you'll see in just a second why I've done that, but I always set it up that way every single time in order to run the gel in the correct direction. So our gel is in the tank. I'm going to add TBE buffer to it. And I'm adding this buffer such that it completely covers the electrodes and completely covers the top of the gel. So I want to make sure we're completely immersed and that we have good electrical continuity. And that should do it. We don't want to add too much because it will make the gel take longer to run. So we want to have the optimal amount. And now I'm going to carefully pull the comb out of the gel. I'm going to do that with both hands, and to the extent that's possible, I'm going to try to pull as much straight up and down as I can. Sometimes there is a little suction as I'm pulling it out, and I just want to make sure I don't tear the gel. So carefully, I'm going to pull this out. There we go. And now we have wells that we can load our DNA into. Hopefully you can see those. So to load our gel, I'm going to get my micropipette again. I'm going to get a pipette tip. I'm going to get our sample that has loading dye in it. I'm going to take up 10 microliters. And I am now going to load that into the first well on the left. We're just loading one sample, but on the gel that you'll analyze, there have been samples loaded in each of the first 4 wells. So that's one difference between this demonstration and what you're actually analyzing. But I am going to very carefully here lower the tip of the pipette down into the well and slowly push the DNA sample out of the pipette tip. So I'm doing that very carefully. If I do it too fast, the DNA will hit the bottom of the well and tend to like burst back out. I'm also being very careful not to puncture the edges of the well with the pipette tip. So I'm loading the DNA into the gap of the well, not like injecting it into the gel matrix. Okay, so I've loaded my DNA. I'm now going to clip on the lid that attaches the electrodes back to the power supply. And the way this lid fits on, we have black and then red. And black always indicates the negative pole, red always indicates the positive pole. And we know that our DNA is negatively charged, so it's going to move to the positive pole, meaning that it's going to move to the bottom. So we always want to double check to make sure, or ask ourselves the question, is our DNA going to migrate in the right direction? And in this case it will, because we want it to migrate down through the gel. We have the red positive pole down at the bottom, and that's why I always set this up the same way, so to make it sort of foolproof, make sure I'm not going to make any mistakes. So I'm now going to set the voltage to 110 and the time to 45 minutes on our power supply. And I'm going to press the run button and it's all going to start running. One way that we know that it has started running appropriately, I'm going to see if I can get a good image of this, is that we will see bubbles start to bubble up from the end of the gel right where the negative electrode is. It might be hard to see in the video, but hopefully you can see it. So our gel is now running, our DNA is going to migrate, and in 45 minutes we'll have a GELDA image, and we'll get back to you with that.